Sampling can be broadly divided into two types: probability sampling, also known as random sampling. The other one is the non-probability sampling. So here, it involves calculation of probability. If you're familiar with probability, is just calculation, calculation of chances. Okay, and whereas in non-probability sampling, we do not compute the probability. Also, in other words, we can say that in probability sampling, each and every element has some chances of being selected in the sample. So let's say this is a population, and we are collecting, or we are sampling a small sample. So this is the population, this is the sample. In probability sampling, each element in the population has some probability of being selected in this sampling, but that criteria is not necessarily true in non-probability sampling, and that's the difference. And we'll see some of this, the types in the next few slides. So what's the sampling process in a research environment? What are the things that you do to sample data from population? So first thing to do is to define the population of concern. So what is your population, and what is the available data that we have for populations? You may not have availability of the true population. For example, let's say you want to do the consumption pattern of the world. So you cannot go to each household and get consumption data, right? So you have to define the population that you have access to, and it need not be the entire population or true population. And from there, you specify the sampling frame. So what is sampling frame? It is set of items or events, so possible to measure. Okay, not all items are easy to measure, so that thing has to be specified. For example, there is a population in place, and we want to sample from that population. Like, not all of these elements in this population are accessible. So sampling frame is about identifying those elements which can be, or the elements or the items or the events which can be measured. So you identify that in the second step, and then you specify a sampling method for selecting these items. Okay, so those items which are indeed accessible and you meet your criteria. Then it's about specifying which sampling method to be used to select the items from the population into the sample, and then determining the sample size. Well, it happens exactly the same time as you decide upon the sampling method that is to be used. Then you implement the method, and then you collect data, you review the sampling process to ensure that it is error free. So these are the steps that you follow in the sampling process. Alright, so what is the probability sampling? So probability sampling, as I've already said, is one type of sampling where every unit in the population has a chance. So this is important. Every unit in the population has some chance of getting selected in the sample. And when each of these units has an equal probability of being selected, then it's called equal probability of select, or EPS sampling. Sampling, on the other hand, is where you cannot really compute the probability of selection accurately. Okay, and we'll see some of the examples also from the non-probability sampling. Simple random sampling is a probability sampling, and this is also a typical case where each and every observation or every element in the population has equal probability of being selected in the sample, and that's why it's called simple random sampling. And it's used mostly when the population is small and homogeneous, because it's computationally very expensive. So if the population is small and homogeneous, we go for simple random sampling. And all elements are given equal probability. I've already said that. For the non-homogeneous population, it's not suitable. That means if you have different segments, then it's better to go for other type of sampling, and we'll talk about what type of sampling is suitable when you have different segments. We use stratified sampling there, okay, and we'll see what stratified sampling is all about. But the problem with this type of sampling is that each segment is not well represented. Okay, if you have many segments in the population and you go for simple random sampling, you will find that some of the segments are not well represented in the sample, and that could be biasing your data, the analysis and your results. Systematic sampling, so it involves a random stat, and then you proceed with a selection for every kth element. Okay, I'll straight away go to the picture and it's easy to understand from there. Okay, so these are different elements of a given population. So the first element that we select in the sample is random. It could be the first one, it could be the second one, third one, fourth one, but it's totally random. So we selected the third one, but remember this is the random selection. It could be any other element also. And then you select every third element from there, right? Every third element from there. Okay, so k here is 3, but k could be fourth, or could be 2, 5. Okay, so it can take any number. Okay, so the randomness is about selecting the first element, and after that it's pretty deterministic. Okay, and it's evenly spread. That's one good thing. Stratified sampling. So each stratum or each segment is then sampled as an independent subpopulation. Okay, so basically you segment the population and then use simple random sampling. That's stratified random sampling. So stratified random sampling is nothing but segmentation plus your simple random sampling. Then that's called stratified random sampling. Okay, and the good thing here is that all the segments are well represented in stratified sampling. Okay, so you have proportionate representation in the sample. Okay, if you have, let's say, male and female, so what you do is that you segment them into male and female population, and then you use simple random sampling for male population and simple random sampling for female population, and then you combine so that both male and female are well represented. Otherwise, if your population has a bias, let's say towards male, your sample will have the same bias there. So it's not going to be well representing in the sample of the study. Stratified sampling, here you see, right, we have men and women, and we want to select 3 women and 3 men from out of these 4 elements in the population. And what we do is that we simply segment into 2 different segments, and then we select 3 out of those 4, and it could be any 3. Okay, so the selection of these 3 are totally random. It's just that when we select, we ourselves segment into 2 different different segments. But in modern software, you don't have to do it. If you mention what the segmentation, what the classification is, it's automatically going to do it for you. Cluster sampling. Okay, it's a 2-stage samplings, and what you do is that so you basically select different sample areas, homogeneous sample areas. So here you have population, and you have homogeneous samples. Okay, basically these are clusters. Okay, these are clusters. So the elements in each cluster are homogeneous with each other and heterogeneous with other clusters, and then you select a few of these clusters randomly. Okay, so you cluster them, and then you use simple random sampling. So here the units are not individuals, but the clusters. Okay, that's a difference. However, you might be wondering, how is it different from stratified sampling? Because we also do the same thing, right? We segment population into different homogeneous group, and then we use sample random sampling. How is it different? Well, it's different actually. In stratified sampling, each and every observation or element is well represented, or is is represented in the sample. However, in cluster sampling, we get only a subset of the clusters. Okay, so let's say we have a big population and we have, let's say, about 20 such clusters, okay, in the populations, and we sample 10 out of that. That means the other 10 clusters are not even present in your final sample. Okay, but that's not good. That means some of these segments are not going to be present in the final sample. Whereas in stratified sampling, all segments will be present. Well, some presence in the final sample. So that's the difference. Then quota sampling. So quota sampling is a non-probability sampling. So you cannot properly calculate the probability and so on. So what you do is that the population is first segmented into different mutually exclusive subgroups, whether it could be male and female, whether based on demography, based on so mutually exclusive. You understand what it means, right? It means that there is no overlap between these two populations subgroups. Okay, example could be in a survey you go for 200 females and 300 males between certain age group. Okay, that means you have already taken a decision to exclude certain number of observations, right? So they cannot be in the final sample. Therefore, you're excluding a priori. Before even you start sampling, you are excluding certain number of observations. And it is important in some cases where some segments are underrepresented or likely to be underrepresented. For example, you're doing a survey for consumption of a particular good, and you see that your population has 80% male and 20% female. Okay, and you think that that's going to be a problem. You really want to have a balanced representation in the final sample, and therefore what you do is that you decide yourself how many females and how many males I'm going to survey. I'm going to get data from. You can also use other filtering criteria. So that's when you decide to to make it more expert driven rather than a probability driven. So it's called non-random sampling or non-probability sampling. Convenience sampling, also known as opportunity sampling, accidental sampling, hazard sampling. So you basically go for the set of elements in the populations which are readily available or convenient. Okay, and by the way, this is not a good sampling technique. You cannot draw inference based on a convenience sampling for the entire population, because your sampling is pretty biased. Let's say you do not have resources to go to rural areas for a survey, and you simply reached out to people in the urban area, and then you did your study. That means it's based on your convenience. You are in city, you simply went ahead with people from the city, and you could not go to rural areas for the data collection, and that's called convenience sampling, because you really preferred or you chose convenience over a proper sampling technique. So sometimes it's okay, but oftentimes it should be avoided. Panel sampling. So this is when you select a group of participants and you do random sampling. You could use simple random sampling, for instance, but you continue to do that for several time periods. Okay, and it can be done for both for probability sampling and non-probability sampling. The only specific feature of this sampling technique is that you do it for different time period, for T1, for T2, T3 and T4. It could be year 1, year 2, year 3, year 4, month 1, or quarter, or something like that. It's used for in marketing. You want to see the growth of consumption for a particular group of people. You can also see how the opinion of people is changing over time, right? So that also can be used. So it's basically used in panel data analysis. Panel data analysis, as you probably know, that requires you to have both cross-section as well as time series data. It combines both cross section and time series, and that's where you can use panel sampling, to get a data which can be used in panel data analysis. Thanks.